Hi everyone, my name is Ashley Schwartz. I am a first-year computational science PhD student in the joint doctoral program between San Diego State University and University of California, Irvine. Today I'm going to be talking to you all about my project titled Developmental Deformities Induced by the Ecotoxicological Contaminant Trichlorophenol methanol, or TCPMOH, in zebrafish. I'd like to take this time to acknowledge my amazing advisors, Dr. George of the Math Department and the CSRC, and Dr. Sant of the School of Public Health. So a little bit about what TCPMOH is. It is a novel water contaminant that has been recently found in our environment. It is thought to be a metabolite or byproduct of DDT, which is a known compound to have extreme harmful environmental effects. And lastly, it has even been found in human breast milk. With all this being said, TCPMOH poses a threat to infant development and our environment. My study objective is to use mathematical and computational models to explain the way that TCPMOH affects development. And to do that, I use zebrafish as a vertebrate model for development. Zebrafish are a widely used vertebrate model, mainly because they are completely translucent, as you can see by here on this microscopy image on the screen. When you look at them under a microscope, you can see all their organs develop, which is, helps assessing deformities during that developmental stage. So for experimentation, me and my lab members collected some zebrafish embryos from breeding tanks. We placed them in an experimental group solution, whether that be control or one of the dose levels here. We captured microscopy images each day during the developmental process, and we assessed morphological data and mortality and determined, if the fish was deformed at every time point. So my first goal was to perform image analysis of all these microscopy images to determine the actual interaction between nutrient usage and the overall fish length or the overall fish growth. Nutrient availability in the zebrafish is measured using a pixel area of this yolk sac here, and then overall fish growth is measured using a fish length measurement here. I developed an ordinary differential equation model to describe this interaction, as you can see here. And after a nonlinear least squares analysis, I found that the model explains the control data really well with an R-squared of 0.99. When TCPMOH was introduced into the system, as you can see here by this new graph, there was an increase in heterogeneity between samples, as you can see by the error bars, but the model still described the data quite well. And in fact, we even found that TCPMOH has an increased rate of yolk utilization and a decreased fish growth. This is important because if the fish uses their nutrients too quickly during the developmental stage, this can pose a major issue for them to develop fully because they only have a finite amount of nutrients available at the beginning. So I continued to assess other developmental deformities, and I actually determined a complex network model that links these developmental deformities to determine the overall impact of these negative perturbations on the developing zebrafish. So I developed a complex network model that looks like this, where each of the nodes are a deformity that we saw during the experimental process. We looked at pericardial edema, which you can see the deformed fish on the bottom image, pericardial edema, yolk edema, swim bladder inflation, cranial malformation, a spinal deformity, and also death, which I just don't have an image of. The links here are, of course, going to be an association link. So just as an example, here is an association heat map of the medium dose group, just as an example at a particular time point. The association between each deformity is going to be given the weighted link between the two nodes within the network model. So in total, I actually have 24 network models, 144 independent nodes, 180 weighted links per experimental group, which is 720 weighted links in total. So you can imagine it does get a little bit complicated when we're looking at all the different dose groups. Node importance analysis was done using spectral decomposition analysis, and the spectral radius for each network is shown here on the screen. You can see that TCPMOH exposed samples had an increase in spectral radius or an increase in the association between the deformities. And in the control group, the network didn't identify any node importance or deformity presentation because controls did not experience developmental deformities. Some key findings for the study are TCPMOH exposures increase the incidence of embryonic defects, they decrease the swim bladder inflation, and increases mortality. Our network model indicates that yolk edema is most highly associated with the development of further deformities and mortality. And in addition, our ordinary differential equation model identified that TCPMOH increases the rate of yolk utilization and decreases the growth rate. This is interesting because in both different models, we're seeing an extreme perturbation to the nutrient availability for the fish. All in all, TCPMOH introduces toxicity to the developmental process, so we need to make sure that we are looking out for TCPMOH in our environment and assessing how we become exposed to this. So in conclusion, here are just some summaries from today's study. I would like to take a second to acknowledge my lab members and the following institutions. And yeah, thank you so much and feel free to reach out.